Hello you guys, my name is Klismann Medrano and today is March 16, 2019 and what we're going to present today is the lab exercise 2 and this is for the bacterial enumeration, dilutions, and plate counts. So I do have my materials on hand. I do have a Sharpie and I already have labeled all of them. And the way you're supposed to label them is first I put the initials obviously on the side. My 10-1, 10-2, 10-3, 10-4, 10-5, 10-6. I also labeled my test tubes. And I have instilled 0.25 ml of water into it. I do have my yeast with it. I have let this sit for 10 minutes and I put 2 tablespoons of warm water and I have mixtured as well with it as well. So let's actually begin. I do have my alcohol so I can clean it as much as I can. So let's get straight into it, you guys. So the first thing that you're supposed to do is you're supposed to instill 0.25 ml waters in each of the test tubes, and you're supposed to label them first of all. So let's go straight into it. I did label them, like I said. I labeled them 1, 2, 3, 4, 5, and 6. So after that, we have labeled all of them, you guys. And this, I do have my alcohol, and I did label it. I hope you guys can see it. Okay. Boom. And my still water too. The first thing that you guys want to do after this is instilled, you're supposed to get 0.25, and this is labeled 0.50. 0.5. So you're gonna grab right here is 0.25, and what you're gonna do first, I'm gonna clean it right with alcohol and decontaminate it. I did wash my hands before and with warm water for about 20 seconds, and prior to this I did wash my hands, and I'm gonna wash it before and after just for the video content. Don't want to keep it too long. So now that I have done that and I cleaned it, the next solution is to get 0.25 and add it to each of the vials. Okay, so we're gonna grab 0.25. Alright, now that we got 0.25, okay, we're gonna let that in there, and I don't plan to let it touch. It did not. 0.25, and we're putting it all in the 6. And honestly, you guys, I'm really not trying to touch the test tubes out of the outer ring. I just want to touch what I need to touch as much as I can. I don't want to. So 0.25 in there into all 6 of the vials. And I'm actually I'm not touching any of the vials. I'm trying not to touch any of the vials. I'm only trying to get what I need, and that's 0.25 as needed. Okay, so that's, and I'm putting in test 1, test 2, test 3, test 4, test 5, and test 6. Alright, I believe that is all of them. Okay, now what I'm going to do now is I need to clean this. So obviously we're going to use our alcohol to clean and decontaminate it. We just kind of want to make sure that it's clean so it's sterile so we can utilize it again. So now that it's clean, we are going to mix it so it can dilute together. Okay, we're supposed to dilute it. I'm going to dilute all of them as much as I can. Okay, I'm not touching that. And they are labeled. And it says to dilute it as much as possible. So trying to shake as much, and you can tell it's more of like a cloudy, thickened. Kind of looks like a UTI in my eye. It kind of looks like a UTI, to be honest. It's how cloudy, or apple juice, a little bit darker than that, or horchata. Anyway, so this is test 1, test 2, test 3, and I'm gonna instill the 10, each of them accordingly. So test 1, test 2, test 3, test 4, test 5. This is 1, 2, 3, 4, 5, 6, and this is 1, 2, 3, 4, 5, 6. Wait, this is 6. This is 5. Okay, and I'm gonna instill only 4 drops into each of them. It should be equivalent to 0.25, if I'm not mistaken. So I'm gonna open it up. Okay, I'm gonna go in there. I did clean it. So we're only going to instill 4 drops. 1, Jesus Christ, 4. Okay, 1. This is 2. So oh, you can smell it too. Okay, so one, 2. I only had 2 drops in there. I'm not sure what's going on. Why is it not? So that's 4. Okay, so that's 1, 2, and this is 3. Okay. So now we're gonna pour 4 drops. 1, 2, 3, 4. You can smell it. You can smell it too. It's actually super strong. One, 2, 3, 4. And I'm gonna shake them all after, just to kind of make it a little bit faster. So that's 1, 2, 3, 4. That's 2. Four. Okay, that's 4 in there. Okay. And this is the 6th final one in my agar plate. 1, 2, 3. I need 1 more drop. And if you're asking whether if you should clean them after, you shouldn't have to clean every single one after, because if you really think about it, they all have the same. There are yeast and water, so technically it'd be like sterile on sterile in the same kind of technique, if that makes sense. So you shouldn't have to, but some maybe professional would like you to clean it, because it does make sense because sterile's on sterile. It's like if it was sterile and you're touching another sterile, and these are both containing the same, so you shouldn't have to. Now that you're done with the 0.5, you would like to shake it up and it to instill as much as possible, and then you would check on this in 12 hours, or what like I said, best is 48 hours, because 48 hours you're able to really able to see all of this and see the microorganism grow, instead of waiting only, like, let's say 12 hours. I feel like it wouldn't grow as much. It's like how they do UAs, the CNS, like a culture and look, when they do a culture and sensitivity of like a urine, they wait 72 hours. So it's best 48 to 72 hours. That's my opinion. Some say 48 hours is a lot better. So now that I have this, I will keep this in the environment that it needs for it to grow. So yeah, you guys, that's how. I hope you guys liked it. I am gonna wash my hands before and after, and I am gonna discard this properly, and then I am gonna wash, sterilize this once again, my surface of usage, because in case if it got contaminated by any chance, I want to make sure that I uncontaminate. I uncontaminate it once again. So I hope you guys like this. And I hope you guys were able to learn something from this. Make sure to label everything. I dated it, I initialed it, and also the microorganism of what I had. So I let the yeast for about 10 minutes. I set a timer, so that's how I kind of knew how long I left it in there. It says 10 minutes, and the yeast I used 1/4 of the package, as it said. Had alcohol and my bulb. So, and then that's when you kind of check after. So I would say you guys wait about 72 hours. It makes sense, but 72 hours should be the best results.